Welcome to this particular uh, presentation. Today would be the last day to talk on chapter 9. We will examine today two important concepts that has changed the entire the production of food material in our country. One is called single cell protein production and second is called tissue culture production. These two are very important areas of applied botany, or we can say applied science, which has created a large number of openings for production of edible food, which are also nutritious. So in class 12 biology, this particular topic is discussed so that you will have more options for your career in when you go to higher level of studies. We are again discussing this particular chapter, especially with reference to population explosion and the demands due to expansion of population. The entire chapter is devoted to discuss how we can use modern technology, advancement of science in producing high yielding and disease resisting food materials, which would certainly meet the demands of the Indian population. The conventional agricultural production, like cereals, pulses, vegetables, fruits, etc., may not be able to meet the demand of the modern man. Today, if you happen to go to a vegetable market in a small city like Mysuru, I will find lorry loads of fruits coming. And this is the case maybe if you think of a place like Delhi, Calcutta, Bombay, Bangalore and all, you can understand what is the amount of fruits we require, vegetables we require, cereals we require to feed one particular city. To feed 136 crore people of India, how much of advances we have to do. So the traditional and the conventional methods were of not meeting the demand. Therefore, we have gone to technological processes of producing the food material. There is a shift also from vegetable eating population to meat eating population. Considerably, there is a large number of people today who consume non-vegetarian compared to yesteryears. Now, if you think of the cereals, especially, about 3 to 10 kilograms of plant material or biomass is required to produce 1 kilogram of animal farming, meat from animal farming. That means 3 kilograms of the cereal will be converted into only 1 kilogram of meat. If the number of animals increase, then we must also increase the production of the plants that would be helpful for feeding the animals. So on one side we have to feed the animals, on the other side we have to feed the human beings. For all this purpose, we have a large number of technologies employed in the production of food material in India. And why we say that 3 kilograms or 10 kilograms of the vegetable matter is required to produce 1 kilogram of meat. That means intermediately there will be many food chain consumers, and they would certainly would consume materials. Ultimately, the animal gets only some food, and then we get 1 kilogram of meat from that animal. See, today we are in a position to tap the microbial activity to produce more food naturally. Microbiological technology, especially the new advances in the environmental engineering, where it combines with the technology, is able to tap the resources from the microbial activity and produce more amount of food. and microbes can be made to produce important nutrients also. There just now I have been going through some of the advances in the environmental biotechnology. They say that the research has been now come up to such a level from the sewage material they can extract nitrogen and phosphate using the microbes without any expenditure. That means economically we can produce more nitrogen, we can create more phosphate. Like that, a large number of microbial research is going on to ensure that people get enough nutrients from even from the waste material which microbes can process it and make it clean available for human beings. Now, one of the important things that we have today is what's called single cell protein, and this is a protein developed by means of using the microbes, especially the the microbial activities, microbial cultures. That means you grow microbes, and then either dead or living cells can be used, and their biomass can be used to extract the single cell protein that will be useful to human beings. We shall study that little more in the next few PPTs on the thing. So we say that we can tap microbes. Microbes are freely available in nature without any money. If you even take 1 cubic centimeter of the area where microbes are present, I am told it will contain 10 to the power of 6 type of microbes in that, and there will be abundance, they will be rich, they can do very many activities which a industrial laboratory cannot do. So therefore, we have to find out the methods of tapping the microbes for our food production. Since the population of both animals and plants are increasing, one with the other, we have to find out alternative sources for especially proteins. One of that will be called SCP or single cell protein. For this, what they do is they use the microbes which are grown at the level of industrial scale and to produce the proteins. Especially some of the algae called blue-green algae will produce a wonderful single cell protein that is available in the market today, and they call it spirulina protein, and that will be very, very useful for the purposes of supplementing the nutrients. And that can be produced even from the sewage. Microbes can act upon the sewage and produce proteins, and the large amount of SCP single cell protein is produced from industries today, and today we can get the tablets, very well refined tablets in the market where we can supplement our proteins. These will serve as food rich in protein, rich in minerals and fats, carbohydrates, and vitamins. And this kind of food is called biofortified food. So, what if they ask a question to you? What do you mean by biofortified food? You can say it is the food extracted from the single cell, which will be rich in or vitamins, added vitamins to that, fats are added, carbohydrates are added, and vitamins are added. All that will include what's called fortified food for the consumption of human beings. Such practices like using single cell proteins, etc., reduces the environmental pollution also. Microbes can be employed not only to create food materials but also to clean the environment, especially sewage and other dirty areas where microbes can clean it and they make it environmental friendly. Therefore, my dear friends, try to find out more and more about environmental biotechnology that is available in most of the IIT courses. You can study that. And now even I'm told engineering courses are introducing a portion of biology for studies. Therefore, you should be able to understand the microbial activity much more than what we have done in our school life. I have just given here three images. One of them, right at the bottom of the three, is the tablet form where single cell protein is converted into tablet form, and that is available in any medical shop. You can ask for spirulina tablets, you will get it. That will be rich in vitamins, rich in enzymes, rich in proteins, like that. So it is bio fortified food material. And the other one, the black PPT that is showing to you, is talking about the advantages of the single cell proteins. How in a small in a biological laboratory we can produce such materials in larger quantities. And the second one, where you got four PPTs, will indicate for single cell protein production, we can use fungi, you can use bacteria, you can use algae, and you can use protozoa. That means we can tap the microorganisms to produce single cell proteins that will supplement the nutritious requirement of all the human beings. Today, you can buy them in the market. So you try to explore much more activities of the microbial activities where they can produce single cell proteins. See, of late, scientists are very much interested in tapping the microbes because they are freely available in most part of the area. Some of the bacterial species like Methylophilus and Methylotrophus, these are the two different types of bacteria, which we will not discuss much, except to say that some bacteria are in a position to produce a large amount of biomass in a very short time. Biomass refers to biological material, and these microbes can be exploited. We can grow them, we can exploit them due to microbial reaction with the environmental, with their own environment, where these organisms can extract nitrogen, can extract phosphate, and they will produce the proteins from the waste material. This morning, I was just listening to one of the lectures on biotechnology. They say that there will be plenty of organisms that can be tapped without any difficulty to produce more and more proteins. Therefore, my friends, try to get into some biotechnology course. That will be your future research. I think computer has done enough. Now you have to go for the microbes, find out more and more. If you just think of one square millimeter of a place where sewage is present, in that you may find millions and millions of microbes which they do different functions. So you can tap that, you'll be able to produce more food. Now people are producing the mushrooms. Mushrooms are in great demand. Again, they are also a source of food material. So we can tap the resources of the fungi. We can tap the resources of the microbes like bacteria. All these things, we can use them to produce more and more food for us. The ultimate aim of all these things, what you are studying, is only to know how we can produce more food, quantity of food, and also quality food. That's the whole idea why this chapter is given. So this will give you more what you call freedom to select the courses you would like to go for higher studies. We are coming to the most desired and most relished topic called tissue culture. Tissue culture is one of the very interesting area. We have done research in the class 12 in my teaching days. Some of those students who did some tissue culture during in their 12th practicals today have become scientists. Some of them have become directors. Some of them have become directors. One of them example I can quote would be a director of CFTRI. So all these students have studied tissue culture during their studies. So what is tissue culture? Tissue culture is a plant technology in which we can develop a new plant from one cell or from a tissue, because every cell has the potential of becoming a new plant, and that's called totipotency. It's a concept. They will ask you what is totipotency. We can say totipotency refers to a capacity of a single cell or of a tissue to become the whole plant with the phenotypic and genotypic qualities, and that is the what we call totipotency. So totipotency is employed in the tissue culture. A small tissue is taken and that tissue is grown in a special medium called in the agar-agar medium, giving nutrients. Then we will get very small tissue-like structure called the callus, and they ultimately become small plants called the explants. So these plants can be used as the germ plasm to produce new plants. This method of producing thousands of plants using tissue sometimes is called micropropagation. So one more question for you. What is micropropagation? Micropropagation refers to utilizing the micro level plant tissue material to produce new plants by using tissue culture technique. And since these plants are produced from the vegetative parts, it is sometimes called soma clones, somatic or the vegetative clones. So you try to be aware of these things when you think of going for your higher studies. Please keep this in mind. Engineering course also is introducing now biotechnology courses. I have shown here two images. On the left side to you, where you have a shelf containing at least about 300 small cartoons, plastic cartoons containing plants. These are all the plants produced by tissue culture technique. Now, what is tissue culture technique? In a very simple way to explain to you, go to the next uh, image. In the next image, you have shown that a leaf is taken, a part of the leaf is taken as a tissue, and that is put into a thing called Petri dish. Petri dish is a glass material containing a roundish structure. It contains special nutrient medium called agar-agar medium or any other medium they can take, add nutrients to that. That is allowed to keep in a highly what you call cleaned area where there is no contamination area is there. In such a area it is kept, and that small piece of the plant or a tissue or a single cell will produce callus or some type of a growth. If that growth is allowed to grow, we can get smaller buds like structure. If they're separated and grown again in a medium, they will become plants. So if you combine these two diagrams, we can say by using a small amount of the plant tissue, we can produce hundreds and hundreds of plants having the desirable qualities that what we require. That is what tissue culture does. See, there are different methods of getting very superior plants. One of them will be the meristems. Meristems, about apical and axillary meristems, are those strips of the plant area where the tissues will be very active. We can tap those things and get the those materials from the plant axillary or in the apical meristems, and we can allow them to go for tissue culture. We get plants. So the the superior plants can be done like that. Even scientists would go to the extent of taking a single cell and then after digesting the cell wall, they will be able to isolate the protoplast without the cell wall. And that kind of a protoplast, only with the plasma, can be taken, and such isolated protoplast will be taken from two very superior plants, may be hybridized. They become the zygote, and zygote develops into a new plant. So that's all the latest techniques that are employed in the plant technology. So that will help us to produce more and more and more plants which are having desirable qualities and also high yielding and disease resisting plants. They are the techniques, and these hybrids are sometimes called somatic hybridization because we are not using genetic material. Though I use the word though, genetic material is employed at the level of genes, we are not using the gametes like the pollen and the ova. Therefore, it is called somatic hybridization. These techniques, I hope you read little more, get interest in that, try to go for a research on that area. Here is a wonderful example of where the protoplasts are taken. When I say protoplast, the cell without the outer cell wall in a plant. You know, plant cells have the outer cell wall made up of cellulose that is removed, and you get the protoplast. That means the cell containing the DNA and also the outer plasma membrane with all the content of the cell. So they have taken as an experimental basis, a wonderful activity has been done. They have taken one cell from potato, one cell from tomato. They have fused them and they made them into go for tissue culture, and then they got some small plants and they planted the plants in the soil, and you know what happened? Magically, potatoes were grown at the soil level, and tomatoes were grown at the above the ground level. That means a hybrid which can produce potato at the underground on the root system, and at the stem level they can produce tomatoes. But such experiments only limited to the laboratory use, though they're possible, but somehow that has not been successfully employed. But even today, many researches are going on that, and sometimes they grow the guava plant on a mango twig. These experiments have been done, but they are not very successful. Maybe days will come when we start fusing the cells, we may get into this type of technology of growing plants. We are coming to the end of the chapter and also the end of chapter 9 now. I have a few questions for you. You can try to answer them based upon what you heard so far or what you read so far, or you have to read and tell. Explain what is meant by biofortification. I explained that to you. Which part of the plant is best suited for making virus-free plants and why? For that, you remember the last PPT I told you about the meristems, they are virus-free. And what is the major advantage of producing plants by micro propagation? You must know first what is micro propagation and then tell what are the advantages of that one. Use your own words. Find out what the various components of the medium used for propagation of explant in vitro. I use the word in vitro so that you understand that in vitro means in the laboratory condition, win vivo means in the living conditions. when you do some experiments in the laboratory condition, in the especially petri dishes or in the test tube, it's called in vitro. And if you do it in the natural setting of the soil, they are called in vivo. So try to know these terms and find out how best you can learn these things before you go to next level of your college studies. The usual Thanksgiving, we call it credits. I give my heartfelt gratefulness to NCRT for using the content for this particular presentation. Creative Commons, I have taken a few images. Of course, YouTube, where you are learning, they will be publishing our YouTube creations. We are thankful to them. Thank you for reading your textbook. I would like to thank you for that. And I would like to say good luck at the end of the chapter 9. I wish some of you will become biotechnologists and find out more and more about how you can tap the resources of microbial population, which will be seen in almost every part. They contain wonderfully useful activity. You have to tap it. Thank you.